Hello guys, I am Shubham from ShubhVed Tutorials. In this video, we study about complementation test. Okay, so let's get started with the video. So complementation test is for recessive mutations. Recessive mutation, right? Means in this we can identify that the mutation is in same gene or different gene. Okay, so see, I will just draw here. These are the genes. Okay, this whole one gene and these are two genes, right? One, two. Okay, these are two separate individuals, right? So now, in the first case, we will discuss that if the mutation is in the see this gene one, I will draw here with black. This is a mutation, okay, right? Again, we will draw here in the same gene one, okay. So phenotype is there, mutant phenotype. Phenotype is same, recessive mutation with the same phenotype, both of having same phenotype. Only we want to know that the mutation is in the same gene or different gene. So now this is the same gene one that is having mutation. Okay. Now if we cross this, what we will get? We will get same like these are the genes and we will get here are the mutations again gene 1. Okay. Here are the mutations, right? This is 1, this is 2 gene. Okay. So here we can see mutant phenotype. Okay. Phenotype seen. I will write a mutant phenotype. Here also mutant phenotype, right? And this is also mutant phenotype. Okay, mutant phenotype. Got it? Right? Now we will see different example. What happens when the mutation in different gene? Got it? Different gene. Again, I will draw your genes. Right? Same here. Okay. Now mutation are in different genes. In this individual, mutation is here in gene 1 and in here second individual mutation is in the second gene, right? So this is the cross. Okay. Now what happens? We know this is the mutant phenotype. Mutant phenotype. Here also mutant phenotype. Then in the progeny, what happens? See, now what happens if the one gene here and one gene here. Okay, so this is gene 1, this is gene 2. So here what happens? We get wild type phenotype. Wild type phenotype means what? Normal phenotype. Okay, means genes are complementing to each other. Okay, so this is known as complementation gene. One gene will complement each other, and we see the normal phenotype in the progeny, right? So this is complementation. Okay, so this is known as complementation gene. Now we will see how problems related to complementation test come. So this is a one example. Okay, given below is the result of complementation test for 6 independent mutant strains of peas. Okay, each homozygous for a recessive allele resulting in white flowers. Each box gives the phenotype of the F1 progeny of a cross between the male parent and the female parent. Got it? So plus represents complementation and 0 represents non-complementation. Okay. So how many complementation groups do the 6 mutant strains belong to? Okay. So now what is the solution here? See, you can see this table here, and now we will just see the non-complementation groups here first. See, when, see, this is 1, right, and this 0 is there. See, no complementation. See, right, again here also no complementation, here also no complementation, right, means we can write here 1 3 6, right, 1 3 6. This is the first complementation group. Okay, then next group is what? Again we will see here, this is again 3, this is 6 already written. Now next complementation is here 5. 5 and 4 means you can write here 4 5. This is the second complementation group, right? 4 4 4 5, and that's it. And the remaining 2 is remaining, so these are the 3 complementation groups. Got it? Okay, that's it. This is the answer. Okay, so that's it for this video. I hope that this video helps you.
Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.